Good afternoon. Thank you for this great opportunity to present our work on transcriptomic biomarkers of mortality at this beautiful site. During this conference, there were many great talks on molecular clocks, and there are several pretty desirable properties that these models may have. So one of them is universality. Ideal clock should be applicable across tissues, across species, then it'd be very convenient to test it in different models. And here, of course, there is a great example by Steve Horvath, pan-mammalian clock, epigenetic clock that can be applied across almost all mammalian species. Another property is interpretability. If clock is interpretable, then we can learn more about potential mechanisms behind certain intervention by looking at top features or key subsystems that contribute to the effect. And a great example here would be organ-specific proteomic clocks developed by Tony Viscaray group, by Vadim Gladyshev group, etc. And also, it would be great if the model that we have could be applied across many already published data sets, including single-cell ones, because in this case, we can identify novel lifespan modulating interventions that have been already tested in molecular setting. So we hypothesized that transcriptomic clocks would fit nicely and cover all of these features. To test it, we first collected gene expression data from tissues of mice and rats across different ages. It's more than 3,500 samples from 96 independent datasets. And we trained first chronological clock of just chronological age across different tissues. And you can see here that the quality, the correlation on test set is comparable to established epigenetic and proteomic clocks. But in addition to that, we also collected gene expression data from animals subjected to different interventions that either shorten lifespan, like progeria models, or extended, like rapamycin, caloric restriction, etc. And for all 79 intervention models that we used, we had corresponding survival data from published established studies. Using these survival curves, we could estimate for every sample that we have, we could estimate expected mortality, which is a composite of aging and the effect of interventions. And now we can use this metric as a readout to train the mortality clock, and that's what we did. So this is mortality multi-tissue transcriptomic clock, and it has pretty nice correlation, 0.95 on test set. And to validate it, we applied it to novel data sets that had intervention data, and these data sets were not included in the training set. And indeed, we found that the clock predicts lifespan shortening interventions to have high transcriptomic age compared to age-matched controls, and lifespan extending interventions reduce transcriptomic age. So mortality clocks can be used to identify novel lifespan modulating interventions. What is really interesting here, these clocks were trained on bulk data, but we can test if multi-tissue clocks capture conserved aging-related changes across different cell types and even across different species. So to do that, we utilized Tabula Muri's single-cell collection. This is a collection of single-cell gene expression profiles of mice across different ages. And we used meta-cell procedure for every cell type. We pulled all cells corresponding to a particular animal together, and then we applied clocks to these meta-cells. And it turned out that in many cell types, we observed aging-related dynamics with age. So you can see even stem cells show aging at the level of transcriptome, macrophages, T cells, muscle cells, etc. And the same model was applied to all these cell types. And actually, from among approximately 50 cell types presented in Tabula Muris, almost all of them showed statistically significant increase of transcriptomic age with time. So it means that multi-tissue transcriptomic clocks, they capture not only differences in cell composition, but also changes within individual cell types. And this is very cool. It means that all cells, almost all cells in our body, basically have shared aging phenotype at the level of gene expression. To expand it across species, we also expanded our data with monkey and human gene expression data from tissues. And I want just to highlight one recent paper that helped us a lot with this. It's by Wanhui Liu, who performed RNA sequences of many monkey samples. And using this data for species, we developed first multi-species, multi-tissue transcriptomic clock. And it means that aging-related changes are conserved not only across cell types, but also across species. They can be applied to primates, to rodents, similar to epigenetic clocks. So we are really excited by that. Now let's shift our gears to interpretability. So we asked ourselves if we can define certain components of transcriptome, certain clusters of correlated genes. And using clustering analysis, we found 26 consistent modules of correlated genes across aging and lifespan interventions. And interestingly, when we performed functional enrichment analysis, it turned out that individual clusters are enriched for very specific functions. For example, we have a subset of genes involved in inflammation, subset of genes involved in cell cycle, respiration, etc. And now, using this subset of genes, we were able to develop module-specific transcriptomic clocks of chronological age and mortality. So now we can track aging at the level of individual cellular components, at the level of inflammatory genes, at the level of metabolism, etc. And here, just to show you, we validated our mortality clocks and showed that it can predict both chronological age and effect on lifespan. So it can be used in both settings. So now let's move to some examples. We started from chronic diseases. We collected data. This is basically public data, 9 data sets on different chronic disease in rodent models in mice and rats, including Alzheimer's disease, chronic kidney disease, ischemic stroke, etc. As expected, transcriptomic clocks predicted diseased animals to be older than age-matched controls, with one interesting exception. For hepatocellular carcinoma, actually the clock predicted cancer cells to be transcriptomically younger than control tissue. We were puzzled by that, and we looked at modules that we identified previously, and it turned out that two most contributing modules in this effect, rejuvenation effect of cancer, are modules involved in differentiation and cell cycle. So indeed, since cancer cells have high proliferative capacity and are de-differentiated, at the level of these modules, they look younger. At the same time, same cancer cells had pro-aging effect at the level of some other modules, like inflammation. So it means that the same model, in this case cancer, can have both pro and anti-aging features at the same time. We also found consistent biomarkers across these diseases. I will not talk much about it, but you can see that they are propagated across different models. And we validated their effect to predict mortality in UK Biobank data. It turned out that their protein concentration in plasma is also predictive of human mortality and multiple different diseases. When we moved to rejuvenation models, and Jesse already mentioned yesterday the model of heterochronic parabiosis, when you attach young and old mice together, and using transcriptomic clocks, we saw that young mouse attached to the old one looks older, but after detachment, the effect is diminished. On the other hand, old mouse attached to the young one looks younger, and this effect is maintained even after attachment. Another established model is a model of cellular programming, of course, and here we have fibroblasts of people different ages, and we see age dynamics there, but iPS cells shown in green have much lower transcriptomic age, and actually we cannot distinguish between iPS cells of young and old people, so they are indistinguishable at gene expression level. And final model of rejuvenation is a model of early embryogenesis. At the level of epigenetics, it was previously shown that first approximately 10 days of mouse embryonic development are accompanied by rejuvenation, by decrease of epigenetic age. And surprisingly, not surprisingly, but we were excited to see the same at the level of transcriptomic clocks. You can see that up to day 10, indeed, we see rejuvenation. And again, it was more prominent for some modules, but not the others. For example, inflammatory model, respiration models showed this effect. But for example, the model of cell cycle or differentiation, they showed aging right from the beginning of embryogenesis. And this example, so this inflammatory module and this is cell cycle module. Surprisingly, these three models of rejuvenations were positively correlated with each other at the level of biomarkers of mortality. And again, the C2 of the same genes that were contributing to the pro-aging effect of chronic diseases, we saw them here, and they're contributing to rejuvenating effects of these diseases. So individual genes may be great biomarkers of both aging diseases and even different models of rejuvenation at cellular and tissue level. So to summarize, we have shown that transcriptomic clocks can be used to capture conserved aging-related changes across tissues, species, and even cell types, similar to epigenetic clocks. We showed that they can be used to identify novel lifespan modulating interventions and to characterize them, and they can be applied to single-cell RNA-seq data. They can be used to identify modules of correlated genes, and we developed transcriptomic clocks for individual cellular components. And finally, they can be used to identify consistent biomarkers of aging, rejuvenation, and diseases. I would like to invite you to read our preprint. It's already available. And of course, I would like to thank our lab, Dr. Vadim Gladyshev, our great collaborators, and of course, you for your attention. Thank you.
Perfect timing. Thank you, Alexander.